Welcome friends. This is the last video in the lecture series of gluconeogenesis and its coordinated and reciprocal control with glycolysis. So let's start. The first slide starts with the question that why PFK, that is phosphofructokinase, rather than the hexokinase, is the pacemaker of glycolysis. So answer to this question is glucose 6-phosphate, which is the product formed by the action of hexokinase on glucose, is not solely a glycolytic intermediate. If not, then what it is? Why it is not solely a glycolytic intermediate? Because it is also converted to glycogen or oxidized by pentose phosphate pathway. You are going to study about these pathways in future videos. So the next is hexokinase isozymes of muscle and liver are affected differently by their product glucose 6-phosphate. So first we need to know what are isozymes. So these are different proteins that catalyze same reaction. Humans have 4 of this kind in case of this hexokinase, and in muscles we are interested in muscles and this liver only. Okay, so in muscles what do we have? The predominant enzyme is hexokinase 2, and 1 and 3 also present. This enzyme in muscles have high affinity for glucose. That is, it is half saturated about even at 0.1 millimolar. So this is quite very less concentration, and this is going to be half saturated at this concentration. So this means this enzyme is normally acting at or near its maximal rate, and this is going to be inhibited by product glucose 6-phosphate. So if it is inhibited, what to do now? So for that, let's move to the liver. In liver, different hexokinase of liver and muscle reflect different role of these organs in carbohydrate metabolism. In order to answer that question, we study a bit more. So muscles use glucose for energy production, that's it, and liver maintains blood homeostasis. So there is a difference between muscles and liver for the use of glucose. How it makes homeostasis? So by consuming or producing glucose, liver maintains homeostasis. The predominant enzyme here is HK4, which is differing from other hexokinases present in muscles in 3 ways. The first is this is half saturated at 10 millimolar glucose concentration. So this concentration is too much, which we studied in case of muscle, which is lower than HK of muscles, you can say. So glucokinase, which is another name of this HK4, phosphorylate glucose when it is abundant, and this is not going to be inhibited by its product, that is glucose 6-phosphate, as we saw in case of muscle hexokinase. So therefore we can say it continues to operate when glucose 6-phosphate is accumulated and inhibits other HK. The third point here is the difference inhibition of HK4. Now we need to regulate this enzyme 2, which is present in liver. Because if this is not regulated, all of the glucose available would be converted. So what would happen if there is scarcity of glucose? We need to send this glucose to other organs, we can say. So we need to regulate this enzyme. This regulation is done by reversible binding of regulatory protein, which is specific to the liver, and this binding becomes much tighter when fructose 6-phosphate, which is an allosteric effector, is present. Then there is one more thing. Glucose is going to compete with fructose 6-phosphate for binding, and causing dissociation of inhibitory protein to relieve inhibition. So there are two scenarios now to understand this case, this mechanism. Okay, the first one is when there is high blood glucose, then what would happen? Glucose would enter hepatocytes via glutamate. So these are specific transporters present on the hepatocytes, that are the liver cells. Okay, and this would activate HK4 by this mechanism which we have just studied, and we are going to see about this more in a picture. What would happen when there is low blood glucose? Fructose 6-phosphate would trigger inhibition of HK4 by regulatory protein. So this F6-phosphate would trigger inhibition of HK4. So HK4 would be inactivated, and this would not be able to convert what? Glucose to glucose 6-phosphate, so that we can avail the glucose for other organs when there is shortage of glucose. So this is a perfect regulation. Let's see more about this mechanism. This is a picture which is showing all this mechanism. So here you can see that glucose is entering from blood into the cell through GLUT2 protein here, and glucose is converted into glucose 6-phosphate through the action of HK4. This is when there is high blood glucose. What would happen when there is low blood glucose? So that is the question now. In that case, this regulatory protein here would come into play. This is going to attach itself with fructose 6-phosphate, and they both combined would sequester this HK4 into the nucleus here. In this way, this HK4 would be inactivated and would not be able to convert this glucose to glucose 6-phosphate, and glucose would be available in shortage of glucose concentration. Okay, so this was all about this mechanism. Let's move to the next slide. So the next enzyme is glycolytic enzyme pyruvate kinase. So there are three isozymes for this, which differ in their tissue distribution and response to modulator. The high concentration of ATP, acetyl-CoA and long chain fatty acids is going to inhibit all isozymes. Liver isozymes, which is the L form, this is the name for this liver isozyme, not muscle, which is the M form. There is additional mechanism for this, that is, it is subjected to further covalent regulation. Covalent regulation means phosphorylation and dephosphorylation. But how it happens? Let's discuss. When there is low blood glucose, what would happen? Glucagon would be released, and it would signal CAMP, that is cyclic adenosine monophosphate dependent protein kinase, to phosphorylate this L isozyme of liver, and it would inactivate this. The phosphorylation would inactivate this L form, and what would be the result? The result is that liver would stop using glucose. Instead, this glucose would be spared for brain and other organs. So indirectly we can say that this is asking for gluconeogenesis.